Hello, everyone. I guess I couldn't have asked for a better introduction than by Maisa. Thanks a lot. And you'll be understanding way better what we're doing thanks to what she just explained to you. What is also great is to have an audience here that can certainly appreciate how minimally invasive surgery has completely transformed the lives of our patients and how to treat them, as you just saw. The thing is, though, that these minimally invasive treatments, they come with the promise of lowering the burden for the patient and the healthcare system by lowering the risk, fewer complications, and a shorter hospital stay. They opened up completely new treatment paradigms, as we just saw, treatment of ischemic stroke, oncology, peripheral vascular intervention. Now, the thing is, there's many, many upsides, but there's also limitations today in order to reach the full potential of these treatments. And in order to show you what I mean, let's go into three examples. So the first one is stroke. I don't have to explain what that means, but basically we have this blood clot blocking blood flow to the brain. And the question that the physician has to ask is, what's the length of the clot? And many, many other features, like what's the composition, what's heterogeneity, is it an embolus or is it actually a diseased wall? Because all of these factors have been shown to actually impact the efficacy of the treatment. And today, as you just heard, this is not possible to estimate. Here's another patient. In this case, it's a blood clot blocking flow to the lower limbs. And the question here is it an old hard clot or is it a soft, fresh clot? Again, the physician can't really figure that out because he needs to know it in order to choose the most effective treatment to actually treat his patient. And so the one thing he can do is talk to the patient, trying to get the medical history, which is a very crude way of evaluating things. And in this case, for example, led actually to choosing the wrong treatment. Now, here's the last example. In lung cancer, what do we want? We want to get a biopsy. To obtain a successful biopsy, we need to put the biopsy needle into the lesion. Where is the lesion? That is the big question. You have to understand, over 70% of nodules at an early stage are in the hard-to-reach outer third of the lung, which leads to up to 58% of cases where, from the biopsy, you actually can't reach a diagnosis. Not reaching a diagnosis means delaying treatment decision, means that the cancer grows while the patient's waiting. Which is why physicians tell us what they actually would like to have is a device that tells them that the biopsy needle was actually in the right spot when they took the diagnosis. So what do all of these treatments have, what all of these situations have in common? Well, we are missing information about the one thing we treat, which is the actual lesion. And that leads to the only thing we can revert to, which is a trial and error approach. And that has actual consequences on the patients. Let's go to stroke. Each subsequent pass that you use lowers the chances of the patient to actually make a good recovery. 60% of the procedures require more than one pass, and hospital costs goes up for these cases for up to 30%. Peripheral vascular intervention. Using the wrong device can actually worsen the potential outcome for the patient. Lung cancer biopsy. A low diagnostic yield means repeat interventions, delaying the treatment, reducing the survival rate for the patient, and increasing the cost of the treatment. So what we clearly see here is we need to go away from this trial and error approach that increases risk and costs and trying to reach first pass success. So managing to get what we want on the first pass to really rake in the benefits of minimally invasive surgery. So how do we get there? We need to overcome this lack of information. And this is exactly what Sensome is doing. We've developed the first connected medical device that integrates our sensor technology, allowing to characterize the lesion while you're treating in real time. To this end, we've built the smallest impedance-based tissue sensor in the world that we are seamlessly integrating with medical devices, and combine this with our interpretable predictive algorithms that allow us, based on impedance, to determine the tissue that we're in contact with. Now, here's an example. The first device to integrate this technology is called the Clotilde Smart Guide Wire for the treatment of stroke. You use it like any other guide wire. You go up there, except that now you are able to scan the clot, and by applying a small alternating current at different frequencies, this information is transmitted using a transmitter to a tablet where algorithms are installed, allowing to characterize the clot while you're treating the patient. So what this concretely looks like, without disrupting in any way the workflow, is here. Physician goes in, he understands what the clot is made of, and he also understands how long the clot actually is. The same technology works for many other indications, like identifying tumors and other tissues. Now, right now, we are in our first-in-human trial. We have treated 31 patients. We had 0 severe adverse events related to the device or the procedure, which is why the DSMB is allowing us to continue the study. We have raised over 17 million euros to date and have recently announced a partnership with Asahi Intech, one of the best guidewire manufacturers in the world, to build the commercial product of our smart guidewire, and obtained FDA breakthrough device designation. We have an excellent team, over 30 people who are motivated every single day, bringing their best from all different domains, be it biology, data science, microelectronics, to make this device happen. Don't let engineers play too much without, of course, having the work hand in hand with the physicians, leading physicians of their respective fields to develop a device that they actually want to use, guided by experienced entrepreneurs on our journey. Now, with this, we can attack very attractive markets. As we've just heard, neurovascular market, many active strategics, early acquisitions in a market that is chronically undertreated. So a huge potential for growth, presenting for Sensome an opportunity of $3 billion. The peripheral vascular market, similar situation, even younger, less consolidated. Again, we are treating just a tiny fraction of the patients that should be treated, with an equally large market opportunity for Sensome. The business model, the way that we actually capture this value, is by licensing this technology to these global players. And so, since 2014, we've been building this technology from the ground up, going from in vitro to ex vivo, all the way into patients. The good thing is that we are expecting to have the results of our first-in-man trial early next year, and at the same time are about to launch 2 new studies looking at the feasibility of our technology in the peripheral and in the cancer indication. Now, you're asking, of course, how can you be part of this revolution? And well, I have a means for that. We are preparing our Series C, targeted for the second half of next year. And if you want to be part of the adventure, then please come and see me. I have a sensor with me, so I can show you how small it is. Thank you very much.